We are beginning today Kingdom Animalia, so we're moving into things that you are a little bit more familiar with. And we start at the very simple. So we're beginning with sponges. So today I'm going to give you an introduction to the Kingdom Animalia, generally what is an animal. And then we're going to spend most of our time looking at one particular phylum within the Kingdom Animalia, Kingdom Porifera, which are the sponges. So very simple phylum, and we're starting with them not because evolutionarily they were the first animals, but because they are the simplest animals. And so we again remember that all animals are just as old as every other animal, because they were all made within the same week. Generally speaking, an animal is multicellular, heterotrophic, eukaryotic, mobile, and has specialized cells. That's the general description of an animal. So, like plants, they are truly multicellular with cell specialization. You have different cells at different part of the body that's doing different jobs. You can't say that about lots of other kinds of life, right? That's part of why a protist can't be a plant, is because they don't have that cell specialization. But animals do. They are multicellular. Your skin cell and your neural cells can't do each other's job. If you were wrapped in brain, you would not be happy. And if you had skin for brain, you would be dead. And so your cells can't do each other's job. You are multicellular, and different cells do different jobs. Animals are heterotrophic, which means they can't make their own food. I've said this a couple times. You can't walk outside in the morning, look at the sun, and reach up towards the sky and go, oh, good breakfast. Okay? It'd be great if you could, but you can't. You are an other eater, heterotrophic. You eat other things. All animals are like that. Okay? No animals are autotrophic. There are some plants that can eat other things, like the Venus flytrap or the pitcher plant or things like that. But definitely the exception to the rule. Generally speaking, plants are autotrophs, and animals are heterotrophs. There are no exceptions in the animal kingdom of an autotrophic animal. Okay. Eukaryotic, we know what that word means. It means they have membrane-bound organelles. Most importantly, they have a nucleus. But they also have vacuoles and lysosomes, and Golgi apparatus, and plasma reticula, all kinds of membrane-bound organelles. Okay? They are mobile at some stage. Every animal moves at some point. Not every animal moves as an adult. The sponges that we'll be looking at in just a moment do not move as adults. Sponges do not get up from one rock and slide over to another rock and decide to stick there. But when they are larvae, they are flagellated and can swim until they fall on a rock that they're going to stick to and grow. So all animals move at some point, and the even inside this sponge, and the whole body doesn't move, there are flagellated cells. There are collar cells that are moving and creating a current of water. So the sponge moves at a cellular level, even if it doesn't move at a macroscopic level. Okay, and they all have cellular, tissue, organ, and organ system specialization. Poriferans don't have organs, but they have cellular and tissue specialization. And once we're done with animals, we'll have organs. Once we're done with poriferans, we'll have organs in the other groups of animals. Okay, so this is an animal. If it is multicellular, heterotrophic, eukaryotic, mobile, and expresses cellular specialization, it's an animal. Okay, so some of these things that we're going to look at, you're like, really, that's an animal? Yes, it is. Moving into phylum Porifera, this is where we're going to spend most of our time. These are the sponges. These are the simplest of the animals. So we're going to start simple, and then we will work up to mammals by the end of the year. We're going to be in Kingdom Animalia for the rest of the school year. And so we're going to be taking our time going through all the different phyla of animal life. We're going to dissect lots of them. And so here we go with that. We're going to start simple. Sponges. They are asymmetrical. If If you look at a sponge, there's no plane that you can divide the creature and have two mirror image halves. Sponges are asymmetrical. Some of them are basically round, right? If you look at especially these chimney sponges, you can look, end on one of them, and it's round, but it's not perfectly like radially symmetrical. If you slice it, you're not going to get mirror halves. It's just kind of round and squishy. Okay, there are people like that, but anyway, the sponge is asymmetrical. It is an invertebrate, obviously. Sponges do not have a backbone. It'd be a really weird sponge if it had any bones at all, especially a vertebral column. But this sponge, they're invertebrates. They don't have a spinal column. They don't have a backbone. They don't have a brain, for that matter. They have a central body cavity surrounded by a porous body. So each of these tubes is an individual sponge, and this is a colony of sponges that are all growing next to one another. And in all likelihood, all of these sponges came from one swimming larva that landed on a rock and started growing one of these tubes, and then the tubes bud off of each other, and they can asexually reproduce children near them. So quite likely these are all clones. Maybe it's a couple of different embryos that landed, but it's likely all clones there on that rock. They're invertebrates, and their body cavity, or their body, is a tube. It's really clear to see that these are a tube. Some of the other sponges we'll look at don't necessarily look clearly like a tube, but they still are a tube, even if it's a short, squatty tube. Okay. Central body cavity surrounded by a porous body. And then we have some terms here for tissues that you will find in a sponge. Most of the body is mesenchyme. Mesenchyme, the CH is a hard sound in this one. So not mesenchyme, but mesenchyme. A non-cellular matrix of supporting spicules and empty space. Spicules are actually made of silica, like glass, right? Glass is silica. And they absorb the silica from the water. There's a lot of dissolved silicate in ocean water because of all the sand underneath and all the minerals floating around. They absorb silicate out of the water, and they create these glass sort of little rods, and they build a scaffolding out of these glass silicate rods. And then their body tissue grows around that glassy sort of skeleton. And that matrix of glassy rods and mostly empty space is called mesenchyme. And then their cells actually live within that. So if you've ever seen a building either being built or repaired and they put the scaffolding around it, and then there you see people crawling around on the scaffolding, that's kind of a good way to picture a sponge. The mesenchyme is this mostly empty space with some scaffolding, and then the cells are like the workers that stick themselves into that matrix and are able to do their little cellular job supported by this scaffolding. Okay, so that's kind of a good mental picture for what we have here. The internal surface of the animal is lined with flagellated cells called collar cells, because when you start to see pictures of this in a moment, the cells have a collar. They've got like a little flange sticking out around the flagella. So they're called collar cells. And they flip their flagella around and move water through the central cavity. So a sponge filter feeds by filtering ocean water through the body of the sponge and out the central hole. So this hole is the waste pore, right? What it doesn't want, it lets flow out that hole. And it grabs little food molecules that come through the body wall. And the collar cells are what create that current. And you'll see near the end of the lecture, I've got a little video of a diver injecting ink around the sponges, and you can see just how strong that current is. The sponges look like they're just sitting there being totally stationary and inactive. But the collar cells actually create a very good current of water that goes through the sponge. So the collar cells create a current of water that moves through the body and out the central cavity. And then the way they eat is weird. There is no stomach in a sponge. They're filter feeding everything. And so if food gets stuck in the body of the sponge, it needs to be able to eat that food. But there's no mouth, there's no stomach, so how does it eat that food? Well, there are amoebocytes, which are exactly like an amoeba. It's weird how much they resemble an amoeba. And these amoebocytes are like mobile stomachs for the sponge. So as the food gets stuck somewhere in the body of the sponge, these mobile stomach cells, the amoebocytes, crawl through the mesenchyme like construction workers climbing ladders and walking around the scaffolding. And they find food particles. And then they digest the food particle. And then that amoebocyte spreads that digested biomolecules around the body of the sponge. So imagine in this picture, you have a building that's being refurbished, and so the scaffolding is all around it. You've got construction workers working on the building, right? And those are the cells of the sponge in the mesenchyme. And it's time for lunch. And one construction worker finds a crate of hamburgers on the scaffolding. And he's like, oh, I got lunch. So he's the amoebocyte. He goes over to the crate of hamburgers. And he picks it up, and he passes out the hamburgers to the rest of the construction workers. That's kind of the job of the amoebocyte. It finds food particles stuck in the mesenchyme, and it passes it out to the rest of the cells of the body of the sponge. Now, to be a perfect example, you wouldn't just hand the food molecule to the other cells, right? The amoebocytes actually digest it and then pass digested biomolecules to the other cells. So to be a better picture, the one construction worker that finds the crate of hamburgers eats them all, and then goes to the other construction workers and coughs up some in their mouth. Okay, so that's the better picture, but it's kind of gross, and you don't want to think about that too long. So handing hamburgers out is a better way to go. But that's kind of what happens. So the sponge is lined inside and out with epidermal cells which protect it. So epidermis is skin cells, right? Same kind of concept here. And these epidermal cells just provide a lining and keep the insides in and the outsides out. Okay. One of your last slides of words, and then we've got a bunch of pictures in the video. Collar cells push water out the top of the central cavity, which causes water to flow through the body of the sponge from the outside. So the current is generated in that central cavity. There aren't collar cells inside the body of the cell moving water through. All the water inside the cavity is kicked out the top, which then creates a current, causes water to be drawn through the body of the cell, through the body of the sponge to the inside. And it's that filtering of water through the body of the sponge that traps food particles and gives the amoebocytes something to find and eat. And then the sponge filter feeds on that current of water, and it eats plankton. Okay. Some of these sponges are really, really beautiful. Some of the sponges here, if you guys go snorkeling, if you take me up in that bodily kinesthetic, and you could get in the water sometime before it's dark this week, there are some gorgeous sponges out here. Some really bright green and red and orange sponges, and some huge examples of sponges. So yeah, if you can find the time to go do that, then you'll be glad that you did. I may even, if you promise me that you're going to go do that this weekend, I may extend the packet due date for those who say I'm going to go snorkeling on Saturday or whatever, and I could give you till Monday to turn that in. But if you turn it in on Monday, it better have that assignment. So then, yeah, some of these sponges are beautiful, and we have some gorgeous ones off of our coast here. So I'll show you some pictures of of sponges here and elsewhere. Okay, so they filter feed on water going through them. This obviously is the background we've been looking at. These are tube sponges. So water is coming in from the side and coming out the top, and each of these tubes is an individual. So I don't know which one of these was the one that cloned itself into all of these other ones. Maybe this one looks like it's kind of taller than the others, might be older. But these are all cloned, budded individuals. Here's just a diagram of what we've been talking about. These collar cells line the central cavity, and you can see why they're called collar cells. Here's the cell, here's the flagella, and it's got this like flange around the flagella, so they're called collar cells. And they spin their flagella and create a current of water. So all the water in here is pushed out the top, and that causes water to flow through the body of the sponge. And while it flows through the body of the sponge, it's filtered out. And these, here they're drawn like little blue blobs, but these little amoebocytes crawl through the body of the sponge looking for food molecules. When it finds something, it digests it. And then as it's crawling through the body of the sponge, it shares its biomolecules with collar cells and the other cells of the body. So it's mobile stomachs crawling through the body of the sponge. So you see that we do have cellular specialization, and we do have tissues, because the epidermis here is a tissue, and the collar cells are a tissue. The amoebocytes don't compose a tissue because they aren't next to each other, all working together as a layer. But they are kind of a mobile, diffuse tissue that spreads itself out through this body of the sponge. So we have cell specialization, we have tissue specialization, we don't really have organs. But the other animal groups will. Okay? Here's more pictures. So some sponges have only one layer of complexity, where there's a wall and a central cavity, and the water flows into the one central cavity and out the top. Some sponges get more complicated, where there are actually side cavities off of the main cavity. So here, water flows in and into this cavity, where then it's pushed out into this cavity and then out the top. So this is a compound sponge, right? This has two layers of complexity. Most sponges, though, that you will see here are not tall and skinny. They're low and squatty, and they're actually this kind of sponge, where they have lots of pockets of central cavities that feed into a very small central cavity where the water leaves out the top. So the red here is the collar cells. In the simple sponges, the collar cells line the central cavity. In this intermediate level of complexity, the collar cells line the side pockets and just squirt all of their water into the central cavity, which then forces it out the top. And in these ones, the collar cells line these central little like bubble cavities, and the water is sucked from here and squirted into the next bubble cavity until it finds its way to the middle, where it comes out. Yeah, kind of like a maze. So obviously, this filters only once. This can filter at a couple of points. This can filter very, very well. So you find these sponges in waters where the food is not as plentiful. And these do a better job of filtering the water. What comes out the top is pretty much just pure ocean water. And all of the little dissolved things that it might have been able to eat have been caught. And it will try to eat them all. So this is a more complicated, more thorough body plan. And this is more of what you find in our water. So that's something that you will see here. Okay. Here's some pictures of sponges. This purple thing is a sponge. It's gorgeous, and it's kind of squishy, sort of, but rigid at the same time. So this is a picture of a purple sponge, and you can see here's a tube and here's a tube, and it's not a smooth body wall. This has got a convoluted body wall. So this is at least 2 layers of complexity, maybe a third. This is another tube sponge. We've been looking at tube sponges as the background of our slides so far today. This is a brain sponge. Isn't that cool? So the central cavity here is very small. If you were to see a picture of one of these from the top, there would be a central hole where all the water is leaving, but the central hole might only be 2 inches across, and this whole thing can be 3 or 4 feet across. So it's sucking water through all kinds of chambers into lots of little tiny pockets with collar cells, and the water is jetting out the top. From all the water coming in through this whole huge big body, there's just a little tube of X current body cavity.
Is that found in Hawaii?
Yes. Yep. We have brain coral. We have brain sponges here. They don't. I don't know if they get this big. They may. And then here's the video I told you about. It's really cool.
Coming up next on Jonathan Bird's Blue World, Jonathan explores the biology of sponges, and you might be surprised at what he finds.
Go, Jonathan.
Hi, I'm Jonathan Bird. Welcome to my world.
Oh my
Those are so fun.
When people think of a sponge, they usually imagine something like this. A cleaning sponge for washing the dishes. Or for washing the car. Cleaning sponges are synthetic these days. But they used to come from the ocean. Back in the days before we had synthetic sponges for housework, cleaning sponges did come from the ocean. They were harvested by hard hat make a dive with a syringe filled with a non-toxic dye called fluorescein. By squirting it around the base of some sponges, we can observe how the water is moving by watching what the dye does. Within only seconds, the dye is pumped through the sponges along with the water.
Isn't that cool?
As you can see, a sponge is a pretty good water pump and also a good strainer. Any plankton that goes in with the water won't come back out through the osculum. Tube sponges are even more spectacular to observe. They pump the dye so furiously that they look like a collection of miniature smokestacks. The ultimate test is a hefty barrel sponge. What will a big monster like this do? It takes a few seconds for the dye to work its way